Hi guys, we're going to talk about the hormonal regulation of glycogen metabolism. And this really builds on the biochemical and the enzymatic regulation of glycogen metabolism. So make sure you review that first. So we need to talk about the two hormones that are most important for glycogen metabolism. And that's glucagon and insulin. So let's start up here. If we have high blood sugar, such as just after we've eaten, this is going to be perceived by the pancreas so that the pancreas releases insulin. And the insulin will travel through the bloodstream to other cells and tissues in the body, like into the muscle cells or into the liver. And in both of those cases, insulin is going to stimulate the formation of glycogen. So it's going to pull glucose out of the bloodstream and form glycogen here in the muscle cells or in the liver. That's going to have the net effect of lowering blood sugar. Well, now our blood sugar is too low, or maybe we've been fasting for a long time. If we have low blood sugar, that's going to be perceived by the pancreas and it will release glucagon. Glucagon is also released into the bloodstream, so it can be perceived by other tissues, such as the liver. Glucagon will stimulate the breakdown of glycogen into glucose. This glucose can be released into the blood and it raises blood sugar. So we're going to talk a little bit more about the details of those two hormone signaling pathways. First, we've got insulin signaling, and the insulin receptor is a receptor tyrosine kinase. So you can see we've got insulin binding to its two-part receptor here, and this is a kinase. So that means it's involved in phosphorylation. You can see when insulin is bound, the receptor autophosphorylates, and then this phosphorylation, this activation of the receptor is able to activate through phosphorylation other protein substrates within the cell. Here, IRS stands for insulin receptor substrate, really creatively named, but it could be a number of different proteins that set up a phosphorylation cascade to activate protein kinases. Ultimately, these protein kinases are going to phosphorylate glycogen synthase kinase. So as noted here, glycogen synthase kinase, when it's unphosphorylated, is active, green for go. This is the active kinase. And when glycogen synthase kinase is active, it will phosphorylate glycogen synthase A into glycogen synthase B. So glycogen synthase B, the phosphorylated version, is the inactive version of glycogen synthase. So when glycogen synthase is phosphorylated, we have an inhibition of glycogen synthase. But I just told you that insulin signaling is going to phosphorylate and inactivate glycogen synthase kinase. That means we're not going to be able to inactivate by phosphorylation glycogen synthase. Instead, glycogen synthase will stay in the active form, the A form, and promote glycogen synthesis. Also part of this pathway, if glycogen synthase A is active and it can be inactivated by phosphorylation, it can be reactivated by the action of protein phosphatase 1, and we'll talk more about that in a little bit. So insulin signaling ultimately through a chain of phosphorylation can prevent the inactivation of glycogen synthase and therefore allow or promote glycogen synthesis inside the cells. Glucagon signaling, we're going to talk about in a little bit more detail than insulin signaling. Glucagon signaling, as well as adrenaline or epinephrine signaling, involves a G protein coupled receptor. And G protein coupled receptors are 7 transmembrane domain proteins. You can see 1, 2, 3, 4, 5, 6, 7 transmembrane domains. And this is called a G protein coupled receptor, or a GPCR, because it is linked to or coupled to a protein that binds GTP, a G protein. When whatever signaling molecule binds to this G protein, that causes the exchange of GDP for GTP. And when GTP is bound, then the G protein becomes activated. And the alpha and beta gamma subunits of the G protein can go on to have other cellular activities. We're going to talk about what some of those activities are. Importantly, the G proteins can be inactivated by GTP hydrolysis. And this can be something that happens just naturally over time, the hydrolysis of GTP to GDP. Or it can be something that can be enhanced or influenced by drugs or other proteins in the cell. So let's talk about some of the details of G protein coupled reception of glucagon and adrenaline or epinephrine. So here I'm just summarizing, I believe, epinephrine signaling or adrenaline signaling. We've got an adrenergic receptor, but this is going to look just like glucagon signaling, except for glucagon signaling will have its own specific G protein coupled receptor. It'll lead to the same intracellular pathways though. Right, so we've got either adrenaline or glucagon signaling here binding to its receptor that causes the activation of the G protein. See, it's binding GTP here. And this active G protein can go on to activate adenylyl cyclase. Adenylyl cyclase has a pretty telling name, cyclase. It's going to be making a circle. Adenylyl, this has to do with the nucleotide adenine or the base adenine. So in this case, we're taking ATP and making it cyclical. We're turning it into cyclic AMP. Adenylyl cyclase, activated by the G protein, is going to produce cyclic AMP. Cyclic AMP is an example of a second messenger. So here on the left, we've got regular ATP, and then with the help of adenylyl cyclase, we can turn this into a cyclical molecule. Look how this ring structure is formed, cyclic AMP. And cyclic AMP is a really common second messenger, especially for G protein coupled receptor pathways. Cyclic AMP inside the cell is going to bind and activate the protein kinase A or PKA. And it's shown here as one easy step. The presence of cyclic AMP activates PKA. If we look a little bit more closely at what's happening, protein kinase A works as a tetramer. So it's got 4 subunits. We've got the regulatory subunits in yellow, and then the catalytic or the kinase subunits shown in blue. And when the regulatory subunits bind to cyclic AMP, they change conformation, they dissociate from the catalytic subunits, and now these catalytic subunits are free to go on and do kinase activity to phosphorylate other substrates. So cyclic AMP has been freed. It's now, or cyclic AMP has freed the PKA. It's now active to go and phosphorylate proteins. And you can see we've got PKA able to activate phosphorylase kinase by phosphorylating it. And again, this is just showing a really simple version of phosphorylase kinase, but you might remember that phosphorylase kinase is a really complex molecule with 4 different subunits. And there are multiple places where phosphorylation of this protein can improve the activation of phosphorylase kinase. Active phosphorylase kinase is going to go on to phosphorylate glycogen phosphorylase into from the B form to the A form. The A form is the active, catalytically active version of the molecule. And again, it's a simplified version here, but you remember your textbook shows phosphorylase A being a dimer that has 2 phosphorylated residues, one in each of the proteins. And this active phosphorylase A is able to bind glycogen and break it down into glucose 6-phosphate. G protein coupled receptor signaling. So that is when we perceive adrenaline or glucagon in our tissues, whether it's the liver or the muscle, glucagon is going to be especially in the liver, adrenaline is going to be especially in the muscle. We're going to be able to increase energy by breaking down glycogen into glucose so that we can go on to do cellular respiration, glycolysis, fermentation, or we can release glucose into the bloodstream from the liver so that all the other organs can have a higher concentration of glucose to work with. Now, we do need to turn off this signaling at some point, and one way that that can happen is through the action of phosphodiesterase. Phosphodiesterase is an enzyme which can break the ester bond here in cyclic AMP, producing AMP. AMP is not able to activate protein kinase A. So the presence of phosphodiesterase means we're going to block this signaling through cyclic AMP and down the phosphorylation cascade. Phosphodiesterase can stop the signaling of this pathway. The next thing I'd like to show you is another related G protein pathway. And this can occur thanks to the binding of glucagon. This is something that happens only in the liver. It's not something that's going on in the muscles. So glucagon can bind to a G protein coupled receptor. And in this case, our activated G protein is not going on to bind adenylyl cyclase. Maybe another G protein is doing that, but this G protein is going on to activate an enzyme called phospholipase C. Phospholipase C, so ACE, we've got an enzyme here. Lipase, this is going to be breaking down a lipid. It's going to be breaking down a phosphorylated lipid. That's exactly what happens. This phospholipase C will break down inositol phospholipid into two components, diacylglycerol, which stays in the membrane, and IP3, which can travel through the cytosol. And that signaling leads to the activation of another protein kinase C. And it also, thanks to the help of IP3, can lead to the opening of calcium channels. So calcium can be stored in the endoplasmic reticulum. We can get opening of calcium channels, PKC, and calcium is released. Awesome. This happens in the liver in response to glucagon. But like, why bother? What is the point of adding calcium to this system? How does calcium even play a role in the metabolism of glycogen? And remember, in this case, we've got glucagon signaling. We want to increase our blood glucose. We need to actually be breaking down glycogen in this case. So when there's calcium present inside the cell, this calcium can bind to and activate the calmodulin subunit of phosphorylase kinase. So phosphorylase kinase, recall, has phosphorylated domains, so they can be activated by pKa, as we just talked about. These phosphorylated domains can also be phosphorylated by protein kinase C. So that's really helpful. We've got two kinases that can activate phosphorylase kinase. And the third way of activating this phosphorylase kinase, which is then, of course, going to go on to activate glycogen phosphorylase, it needs to be binding calcium. So binding of calcium in this calmodulin domain, as well as phosphorylation, is going to lead to full activation of phosphorylase kinase so that we can fully activate phosphorylase and start breaking down glycogen into glucose at a very quick rate. But wait, there's more. G protein coupled receptors producing or activating adenylyl cyclase, producing cyclic AMP, activating pKa, can have effects other than influencing the activity of protein or phosphorylase kinase. In this case, I want to talk about protein phosphatase 1. You might recall that protein phosphatase 1 is important for inactivating phosphorylase and, yes, activating glycogen synthase. So When it's active, we're going to be promoting glycogen synthesis. But if we're doing adrenaline and glucagon signaling, I don't want to be synthesizing glycogen. I want to be breaking it down now, releasing that into the bloodstream to increase our blood sugar. I need to inactivate protein phosphatase 1. So the activity of PKA can phosphorylate a subunit or a coworker, a complex protein with PP1. And this breaks apart or changes the conformation of this subunit so that PP1 is no longer bound to it, and that decreases the activity of PP1. And additionally, PKA can phosphorylate and activate this protein phosphatase 1 inhibitor. So now we have doubly inactivated protein phosphatase 1. It's no longer active to promote glycogen synthesis. Instead, we're going to be blocking glycogen synthesis and allowing glycogen breakdown to occur. So just to clarify, how will PP1 inactivate? How will the inactivation of PP1 affect glycogen metabolism? If we have active glycogen synthesis, if we have active PP1, we're going to have active glycogen synthesis. In the presence of glucagon, that's not what we want. We want to be breaking down glycogen. And so this is inactivation of PP1 will shut off glycogen synthesis and allow glycogen breakdown to begin to restore cellular blood glucose levels. So that's all I wanted to tell you about G protein signaling. There's a lot going on here. There are a lot of different pathways, but there are many conserved types of pathways. And though we're talking about adrenaline and glucagon signaling or insulin signaling, these are similar signaling pathways to what you would encounter for other receptor tyrosine kinases or for other G protein coupled receptors signaling pathways. It's just that ours, or the ones we're focusing on today, are nuanced because they're influencing glycogen breakdown within the body. So send me questions if you have any. I'm happy to chat more.